All right, welcome everyone. I'm super excited about this class tonight, like I'm sure everyone else is. If you've been here and saw the class that we did yesterday, I know you're excited as well. So we had a great class yesterday. I really enjoyed it on the mitochondria and exactly what the mitochondria does, what it stands for. We dealt more so with the atomic. I apologize, that's my phone. We dealt with the atomic structure of the universe. Right? So, quick recap on that. We dealt with the atomic structure of the universe. We then went into the understanding hydrogen itself, helium, how the earth and world was created, right? Because we want you to understand and have a respect for the creation of the mitochondria itself. We went from that into understanding atoms and how they work, how the stars and what they do, breaking down um, into the chemical compounds which are creating molecules, and then from molecules being created, we went into the prokaryote, eukaryotes, and that is basically where we ended last night. And so it was a full class. It was an excellent class. Prayerfully, you got a lot out of it. And we're going to pick up from there dealing with the mitochondria, right? And so I wanted to show you and give you a total respect of how the body was made, not just from a structural standpoint, but also from a universal standpoint, from the hydrogen down. So you could understand, or hydrogen up. So you could understand the actual structural elements of the universe, how they came together, the perfection, the fine tuning of how the earth was sitting on a knife's edge, or how the universe is sitting on a knife's edge of perfection. If it went this way a little bit or that way a little bit, like 0.04%, that there would be no life in the universe. And as a matter of fact, all the hydrogen would have been used up immediately after what they called the Big Bang. And so, again, as I stated, I don't believe in evolution, I believe in creative evolution. And that there was a time process of learning to create the next level of each level of information. So, I do believe that. So, there was a creative, that's why there always is what they call a missing link. A missing link just means that creation came in that step, and they had to it was the next level of creation. See, if evolution were true, just pure evolution, then apes would still be turning into human beings, or Elohim really, but human beings. So apes will be turning into people. That doesn't happen. Therefore, we know that the theory of just evolution is not true. It was creative evolution. The conscious created at different stages of development, but it had to create each stage of development, starting with hydrogen to helium to lithium to what no, not boron, to yeah, boron, to what's the next stage after that, beryllium, so forth and so on. And if that's why the periodic chart is as beautiful as it is because it shows you how creation was created in order when it came to the atoms. Then we dealt with the molecules. Now we're going to be dealing with, we went all the way to the prokaryotes, the eukaryotes, and now we're going to be dealing with the organ called the mitochondria. Okay? And so we're going to start there. If you missed a class from yesterday, I advise you to go get that class. It was excellent. It's called the Mitochondria Part 1. Watch it while it's available on YouTube. If not, you're going to have to watch it. You're going to have to be able to get the with the YouTube subscriber, or the or a health class subscriber on my Patreon, patreon.com/health class. You can get all my health classes and everything there for a small subscription. So you're welcome to do that. Also, I want to announce that tomorrow we will have the creation series again. So for those of you that have been following along with the creation series, it's an outstanding opportunity to take advantage of that. If you have not subscribed to our YouTube channel, you will not be able to chat. So prayerfully that you can subscribe so that you're able to tap into the chat. Okay. So I appreciate everybody on that. So what I'm going to do now is I'm going to transition just a second. I have to give the disclaimer to the testimonial, and then we'll be right back.
Disclaimer. The information contained in this video is intended as an information resource only and is not to be used or relied upon for any diagnostic or treatment purposes. The video's information does not create any patient-physician relationship. Dawood Yisrael makes no representations about the accuracy or suitability of this content, as scientific and medical information updates and changes daily as more substantial information is discovered. Dawood Yisrael and any business associated during this video shall not in any event be liable for any direct or indirect damages, losses, injuries, or liability whatsoever suffered as a result of your use or misuse of this video's content. Use of this video is at the viewer's sole risk.
Go ahead.
Being here is he's been bigger than I could even articulate. Has chosen to share a gift that he's been blessed with. I already knew I was the most ignorant person, but this brother never judged me. I came clean with my errant ways, never felt judged, and I'm leaving feeling better on so many levels. And I'm so thankful for him and his peers that showed up. So I'm looking forward to being back next week to gain some more knowledge, and I can pass that on to my grandchildren and hopefully anyone in my circle. But I'm going to grow with him if he allows me to, and I'm grateful.
Absolutely. You grow with him. All right, guys, we're going to go ahead and get started. Prayerfully, you can see and hear me. Right? Put a 1 in the chat if you can see or hear me, please. I greatly appreciate it. But we are going to go ahead and get started. So how was the mitochondria created, right? So this is where we kind of left off yesterday with eukaryotes, right? The eukaryotic beings, eukaryotic beings. And so we're eukaryotic, right? Bacteria is prokaryotic, as we ended off on yesterday, right? And so the single cells are prokaryotic. The multiple cells are eukaryotic beings, okay? And so we are eukaryotic, okay? So is plant life, so is fungus, all right? They're all eukaryotic organisms. So according to the widely accepted endosymbiotic theory, now this is their theory, right? They don't know how the mitochondria was actually created or developed. They don't know, but this is the theory that they came up with, right? So I just want you to be clear. This is not a fact. It's a theory, okay? In other words, an opinion. I want you to be very clear on that. So according to the widely accepted endosymbiotic theory, mitochondria originated when a primitive eukaryotic cell engulfed an aerobic prokaryote, right? Bacteria capable of using oxygen to produce energy, which means if that's the case, then the original eukaryotes did not have mitochondria. That's the only way this could be true. Now, I've never seen any readings or writings where they said that a eukaryotic entity did not have a mitochondria. However, for this to be true, it couldn't have, right? It had to develop the mitochondria by engulfing an aerobic prokaryote, like a bacteria, or a bacteria capable of using oxygen to produce energy, right? Which then you have to ask yourself the question, how did this primitive eukaryotic being create energy, right? For this to be true. I'm just saying. It could be possible. I'm not saying it's not. I'm just saying this is how they say it happened. But it's a theory, so we can question it real good. So eukaryotic cell engulfed an aerobic, meaning it used oxygen, right? Right, oxygen to produce energy. So that's aerobic. Prokaryote bacteria capable of using oxygen to produce energy, which then established a symbiotic relationship with the host cell, meaning they said we can work with each other, eventually evolving into the mitochondria we see today. So, in other words, this eukaryotic cell had no way to produce energy, therefore, it engulfed or ate or ingested, we could take it that way, an aerobic prokaryote because it needed a way to produce energy, which then established a symbiotic relationship with the host cell, eventually evolving into the mitochondria we see today. Essentially, the host cell gained a source of energy while the engulfed bacteria gained protection and a stable environment to live in. This is what we would call our gut today, right? This is what our gut would represent for bacteria. Okay? Now, remember, bacteria are geniuses. I told y'all that yesterday. Bacteria is a genius. If one day I'm just gonna do a whole class because you need to understand bacteria. Because when you understand bacteria, you'll understand you can never get sick. Viruses don't really exist and all that. You'll learn all of that. I'm gonna do a full class on bacteria. It might be a series because they're ingenious, right? So while the engulfed bacteria gain protection and a stable environment to live in. Okay. Now, key points of the origin of mitochondria. Endosymbiosis. This is the central concept where one organism lives within another, mutually benefiting with each other. Now, we know that's true because we have massive organisms, millions, trillions actually, of organisms living inside of us that we work together with them. And the only way you really get sick is when you throw that off. Prokaryotic ancestor. The engulfed bacteria is believed to be from the alpha proteobacteria group, a type of prokaryote, right? Then genetic evidence. Mitochondria have their own small circular DNA, which they do, which makes them a prokaryote. Mitochondria have their own small circular DNA, which is similar to bacterial DNA, further supporting the endosymbiotic theory, which could be true. I'm not saying that it's not. I'm just saying, where would it have the energy to move, to engulf it, if it doesn't have, if it couldn't produce energy in the first place? That's why I believe in creation. I believe in creation, evolution, meaning at this point, so the first thing that was necessary was to create the prokaryote. Then the spirit could go into the prokaryote. That spirit said, we need something else that is going to give us better mobility. And the eukaryote then was created because the prokaryote could already be put inside of it because the prokaryote had already been perfected. This is, to me, how evolution takes place. That's why I said I believe in creative evolution, not evolution, regular evolution, right? Because if so, again, like I said, apes would still be turning into people, and they don't. So that disproves regular evolution to me straight up. That's the end of that. Over time, many genes from the engulfed bacteria were transferred to the host cell's nucleus, having the mitochondria dependent on the host for most of its proteins. Okay? So that's what it's saying. Over time, many genes from the engulfed bacteria were transferred to the host cell nucleus, having the mitochondria dependent on the host for most of its proteins. Okay. Mitochondria and chloroplast. Okay, we can move past this. All right. So the next thing, different theories of mitochondria. They stick with the endosymbiotic is what they're gonna say. Okay. Now, what is the mitochondria? Right. This is important. Mitochondria are small organelles found within cells. So remember, inside the cell, there are multiple organelles, okay, like the nucleus and so forth. Mitochondria are small organelles found within the cells that are primarily responsible for producing the cell's energy, often referred to as the powerhouse of the cell. Okay, this is what it's called to the powerhouse of the cell. By converting food molecules into adenosine triphosphate, meaning ATP, through a process called cellular respiration. This is a very important term here, right? Even when I'm dealing with the balance oil, we're fixing this. This is how deep we're going. We're going all the way to the DNA of each cell, the mitochondria of the cell, creating cellular respiration. Okay? This is very important. This is super important. This is what? Removing waste and allowing nutrients in. That's cellular respiration. They have their own DNA and are unique in that they are inherited only from the mother. So the mitochondria's DNA is inherited directly from the mother only. So whatever's wrong or right with her mitochondria, the woman's mitochondria is going to make the difference in the child. This is why it's so important for women and men for a different reason, but women for the mitochondria especially, to be healthy. All right? Super important. They have their own DNA and are unique in that they inherit it only from the mother. Function generate most of the chemical energy needed for cellular reactions, storing it as ATP. Double membrane bound organelles with inner membrane that folds in the cristae, right? So these folds in here are cristae in between. Okay? And I'm going to show you this in a minute, so don't worry. I'll show you in a minute. Genetic material contain their own small DNA molecule distinct from the cell nucleus DNA. And mitochondrial DNA is passed down solely from the mother. Those are key points, okay? Now, how does the mitochondria produce energy for the cell? Because tonight we're not going to get into really mitochondrial cell production or energy production. We're going to talk about it, but we're going to deal with glycolysis tonight, which is something that happens before the mitochondria turns it into energy for the cell. But you need to understand it so that you can understand what's going on inside the mitochondria when it's producing energy for you. Okay? So glycolysis happens in the cytoplasm. It doesn't happen in the mitochondria. But the mitochondria does what we're about to read now. Mitochondria produce energy for the cell by converting chemical energy from food into a usable form called ATP through a process called oxidative phosphorylation, where electrons are transferred along a chain within the mitochondrial membrane, creating a proton gradient that derives ATP synthesis. Essentially, they break down glucose and other nutrients to generate ATP, which acts as the cell's primary energy currency. Okay, and they call this process the Krebs cycle. So glycolysis, even though a lot of people will tell you glycolysis happens in the mitochondria, it does not. But the Krebs cycle does, and that's where it deals with the Krebs cycle, and we're going to talk about that in a minute. The process begins with the breakdown of glucose from food into pyruvate during glycolysis. That's what we're going to study tonight. Okay, how that happens. We're going to go deep into that. Which then enters the mitochondria, right? So it comes from the cytoplasm through glycolysis and then enters the mitochondria and goes through the citric acid cycle, also known as the Krebs cycle, to produce high energy electron carriers like NADH and FADH2. NADH is stronger, is more energy than ATP. Just to let you know, it's stronger, right? It's a high energy electron, NADH. Okay, so that NAD is super important. electron transport chain. These electron carriers then donate their electrons to the electron transport chain, embedded in the inner mitochondrial membrane, which pumps protons across the membrane, creating a proton gradient. ATP synthesis. This proton gradient then flows through ATP synthase, an enzyme complex, driving the synthesis of ATP from ADP, adenosine diphosphate. So ATP is adenosine tri, meaning there's 3, right, phosphates. Adenosine diphosphate is 2, right, phosphates. That's it. And ADP is almost like it's almost like a resistor. It's like a resistor. We'll get in that tomorrow more so, but it's like a resistor. I might get into it today because we're going to deal with a lot of ADP. As a matter of fact, we are going to get into that today. In a phosphate group, okay? So oxygen requirements. This is important. Oxidative phosphorylation requires oxygen. And I want you to remember this point. Do not forget this during the class. It requires oxygen. It's an aerobic process. So the Krebs cycle is an aerobic process. Okay? In other words, it requires spirit in order to take place. Okay? As the final electron acceptor in the electron transport chain. Mitochondrial DNA. Mitochondria have their own small DNA molecule which codes for some of the proteins involved in the electron transport chain. Okay? They have their own DNA. Right? This is why they're considered what? Prokaryote. Powerhouse of the cell. Because of their crucial role in energy production, mitochondria are often called the powerhouse of the cell. Super important. All right? Now, mitochondria function in humans. Parts of a cell known as the energy factories. Mitochondria are parts of the human cell known as the energy factories. Most human cells, animal cells, and plant cells. Shalom. Jerry, welcome. Most human cells, animal cells, and plant cells contain hundreds or even thousands of mitochondria. Some cells, such as muscle cells, contain more mitochondria than those that are less active, like skin cells. This article describes the structure and function of mitochondria and explains how they affect your body and health. Okay? All living things are composed of cells. Each cell contains structural components called organelles. Almost like mini organs with specific functions, mitochondria are a type of organelle. Okay? So let's look at this right here. This is what I really want to deal with right here. So the parts of it. So the DNA, you see these little squiggly lines in here, right? That's the DNA. Okay? So you can see that. The matrix, right? You see that right here. It's showing you this is the matrix, right? Inner membrane, right? The inner membrane. The cristae, those are those folds that they were talking about a minute ago, right? Inside. This is the cristae, these folds inside of here, they're saying. And then the outer membrane is this part out here, okay? Now, and then the ribosomes, you remember those from yesterday. So both prokaryote and eukaryotes have ribosomes, okay? Now, all living things are composed of cells. Each cell contains structural components called organelles, almost like mini organs with specific functions. They're not like mini organs, they are mini organs with specific functions. Mitochondria are a type of organelle. Mitochondria are distributed throughout the body. Every tissue and organ in the body is composed of cells, meaning you are trillions of cells. So that's why the cell had to be perfected. So the prokaryote had to be perfected, and the cell had to be perfected in order for you to even exist in this form on the planet. We're trillions of cells. Those are the building blocks. And what is the first building block of everything is hydrogen. Actually, it's quarks. But I'm not going to quantum physics right now. I'm staying in just regular physics. So it's hydrogen. Everything is composed of hydrogen all the way up. So you see the building blocks to create these things. That's why I said it's creative evolution. So now that the prokaryote was created, now the eukaryote could be created. Now we could have bodies that are created from multiple cells. So I don't know, even the quadrillions of hydrogen creates everything more than that, like more than that, right? That creates, and this is how nature builds, or the Elohim build. And so everything has to be perfected step by step by step. And again, if you missed the video yesterday, that's a rewatch. Make sure you watch that and pay close attention. Mitochondria are distributed throughout the body. Every tissue and organ in the body is composed of cells, and most cells have many mitochondria. Some cells have more mitochondria than others due to their energy needs. The body's cells are enclosed by a cell membrane. They have a nucleus which holds the genetic material. I showed you that yesterday. I showed you the whole structure of a cell. And the cytoplasm, which holds the organelles. The mitochondria, like other organelles, are located in the cytoplasm. Okay? So, basic structure. Within a cell, each mitochondria is enclosed by an outer membrane and also has an inner membrane, which we just read about. We looked at, right? What do they do? The main function that occurs inside a mitochondria is the chemical reaction that creates ATP, a molecule that body uses for energy. The mitochondria also have some other roles that are not well understood as ATP production. So they do other things just besides create the ATP, right? And go through the Krebs cycle. They do other things. A mitochondrial function that has recently received attention in the context of health is that of cell regeneration, which involves cell death and renewal. So again, if your senescent cells are not dying off, that means you have a problem with your mitochondria. There's a mitochondrial problem. You got it? This is where xenogene comes in. And you renew what? New stem cells to replace them. Right? Is where water fasting comes in also. It does that as well. Long supervised water fasting does the same thing. So this is going to give a boost back to your mitochondria. Okay? Very important. Energy production is the best recognized and comprehensively studied aspect of mitochondrial function. The structures within the organelles are able to generate ATP through a multi-step chemical process, the Krebs cycle. During this chemical reaction, ATP is produced from glucose, sugar, and oxygen, and carbon dioxide waste is produced, right? The two major processes of this production, the Krebs cycle and oxidative phosphorylation, take place in the mitochondria. The ATP that is produced can be stored in the cell. A healthy cell requires efficient production of ATP to meet its energy needs. This is very important. A healthy cell requires efficient production of ATP to meet its energy needs. And we're going to talk about that tonight when we get into glycolysis. Okay, because we're going to go deep into glycolysis. We're not going into the Krebs cycle. We will do the Krebs cycle tomorrow. Okay? So, cell death, apoptosis, is cell degeneration that is controlled. Cell degeneration can occur as part of a growth and development as a natural part of aging. No such thing as that. But it's not. The cell degeneration is going to take place for cells, but it should give you new cells. If you're not producing new cells, there's a problem. Because remember, if you go back and watch the video on my Patreon called Physical Immortality, where I deal with that, I show you directly through their documentation. That's why I use this documentation, so you know it's not me talking. I use their documentation to show you that there's no reason you can die from old age. It's impossible. You cannot do it. There's always a proximal cause. Anything that is causing you to age to death, to die, versus just maturing, that means that something is wrong. Age can never cause death. That's impossible. There always must be a proximal cause. Something has to show up. Cancer, diabetes, high blood pressure. Even if you think that person just died from old age, it's something you're not finding, but it's a problem. You should never die from aging. That's impossible. Senescence cannot cause death. Additionally, cell degeneration can occur as a result of damage or disease. The mitochondria play a role in programmed cell degeneration and damage, induced degeneration. The mitochondria release enzymes, substances that mediate chemical reactions that lead to the death of the cell, right? So if you got old cells that are just sitting there and they're not dying off and they're not going anywhere, that's going to cause type 2 diabetes, can cause type 1 diabetes, mostly type 2 though. It can cause brain issues, cancer, lung, all of it, because those need new cells there. And the mitochondria is responsible for that. It's responsible for that. The mitochondria are involved in several other functions. This is what in other words, if the mitochondria does not do what it's supposed to do, then what's going to happen is you're going to have zombie cells, or what's called senescent cells. Right? And we dealt with that. The mitochondria are involved in several other functions and can play a role in the initial steps, or may be involved in one of the more subsequent steps of the physiological process. Functions that involve mitochondria include calcium movement in the cell, synthesis. So calcium movement in the cell, that's super important. Synthesis of hemi iron, right? Synthesis of hemi iron. This is anemia right here, right? This is what a lot of sisters run into. Raw food is run into this problem right here. Synthesis of hemi iron, right? Formation of iron sulfur clusters, which help in a variety of biological functions. So if we don't clean up our cells, right? Our blood and our cells, we don't clean that up, the mitochondria is going to have a major problem performing its functions. Okay? What happens when the mitochondria doesn't function like it should? There can be many consequences of mitochondrial dysfunction, including low energy and impaired cell regeneration. Several diseases are known to be caused by impaired mitochondrial function. Leigh syndrome. This childhood neurodegenerative disease are inherited due to the defects in the mitochondrial DNA. This disease is usually fatal within the first 5 years of life. Mitochondrial myopathy. Symptoms of this condition include weakness, muscle cramps, and muscle atrophy, thinning of the muscles. There are several variants of mitochondrial myopathy with different severities of age onset and inheritable patterns. Now we can see why the balance oil actually does what it does and allows the body to heal again. Because it aids in allowing the cell to get back to the point where it can do proper cell respiration. Now the mitochondria can do its thing. Right? Myoclonus epilepsy with raised red fibers. This condition causes seizures, muscle jerks, muscle weakness, and impaired coordination. Right? Mitochondrial diseases are diverse and are estimated to affect 1 in 5,000 live births. Some mitochondrial diseases are inherited. Again, you can always fix that though. The hereditary patterns of mitochondrial disease is very complicated. And if you're getting it, it's going to be from the mother. Why? Because mitochondria is inherited directly from the mother. Hereditary mitochondrial diseases can be passed down due to defects in the nuclear DNA. This is the DNA that contains most of the genetic material of the body. And some are passed down through mitochondrial DNA. One of the features of mitochondrial DNA is that it is inherited maternally, which means that it is inherited specifically from the female genetic parent. Additionally, many diseases that are not specifically caused by mitochondrial dysfunction may involve impaired action of the mitochondria, even when mitochondrial disease is not the main cause. Examples include cancer, Parkinson's disease, and Huntington's disease. These diseases can cause mitochondrial damage, and damage to the mitochondria can worsen the disease. All right. So, right. So those type of things, when it comes to your function, those are more so like inflammatory. Yeah, I guess you would say those are inflammatory diseases as well. They're not autoimmune disease. Let me see. Yeah, no. So with that, we're gonna move forward into cellular respiration. We need to understand that in eukaryotes, because it's different for a prokaryote. Prokaryotes cellular respiration is different than eukaryotes. Okay, so cellular respiration for a prokaryote being, they can just pull it straight out because they're a single cell entity. They can just pull it from the air, from oxygen. But a eukaryote cannot do that. Okay, it cannot. So we're going to learn about how it works. In eukaryotes, cellular respiration primarily occurs within the mitochondria, where the breakdown of glucose molecules generate energy in the form of ATP through a series of steps, including glycolysis. But remember, glycolysis does not happen in the mitochondria. It happens in the cytoplasm. And this is something that you have to be very clear on, because they're two different functions and they function differently, which I'm going to show you in a minute. Where the breakdown of glucose molecules generates energy in the form of ATP through a series of steps, including glycolysis. The Krebs cycle and the electron transport chain. This process is considered aerobic respiration when oxygen is present. Okay? Now, let me see if it says it here. Location. Mitochondria are the primary site for cellular respiration. It is for cellular respiration. That is a true statement. In eukaryotic cells, the main stage of cellular respiration includes glycolysis. Now you see they say in the cytoplasm. You see that? Not in the mitochondria. But I want you to be clear, because glycolysis is anaerobic. It is not aerobic. It's anaerobic. The Krebs cycle mitochondrial matrix, right? That's where the Krebs cycle happens. Because what? It's producing. It's having what? Babies. The Krebs cycle in the mitochondrial matrix and the electron transport chain inner mitochondrial membrane. Okay. Energy production. The primary energy molecule produced is ATP. Most eukaryotic cellular respiration is aerobic. Most. The majority is aerobic, meaning it needs spirit to take place. Right? Or oxygen. Most aerobic cellular respiration is aerobic, meaning it requires oxygen as the final electron acceptor. And it does. It must have that to finalize it. But it starts anaerobic, and I'm going to show you that in a minute. Eukaryotic. As part of the eukaryotic system, are blood cells. This is the question. Are blood cells eukaryotic or prokaryotic? Because we know that blood needs what? Cellular respiration. So because prokaryotic beings can just pull blood out of the air. They can pull energy out of the air. They'll just take air and use that to create their energy, right? I was saying, and they don't, and they're single cell organisms. I was wondering if blood cells were also prokaryotic. But I want you to understand something. But remember, anything that's prokaryotic They do not have cell walls around their organelles. Okay? They don't have cell walls around their organs. All eukaryotic beings have cell walls where everything is separated. Theirs is just in a thing of goo. It can move around. So they're like their lungs and this and that. They can move around in their body. It doesn't sit in one place. Okay? Eukaryotic beings, they got walls. They have determination spots where everything belongs. Okay? So as part of the eukaryotic system, human blood cells are classified as eukaryotic cells. They begin life with cells and mitochondria. So blood cells have a mitochondria at first, just like other eukaryotic cells. But as they mature, they lose their organelles. Okay? I want you to understand that. They lose their organelles. So they lose their mitochondria. Blood cells are considered eukaryotic cells, although mature red blood cells lose their nucleus. They are still derived from eukaryotic cell and classified as such, but they more so become a prokaryotic being. But there's one difference, and I'm going to show you what it says in a minute. Eukaryotic cells have a nucleus and membrane-bound organelles, which is characteristic of animal cells, including blood cells. Red blood cells' peculiarity, meaning what makes them different. While mature red blood cells lack a nucleus, this is a result of a process during maturation where the nucleus is ejected, meaning they kick their mitochondria and everything out of their body. Each blood cell does, allowing for more space to carry oxygen. Now, this is the key point right here. Even though they lose their nucleus, meaning they get rid of their mitochondria, they get rid of everything, so that they can carry more oxygen, red blood cells are still considered eukaryotic. Why? Because they originate from eukaryotic cell. But another thing, and I read this, I thought I had this here, but I don't have it right here. The other reason they are considered eukaryotic still is because they do not have their own DNA. When they kick out that mitochondria, they don't have any differentiating DNA except yours. That's the difference. Or else they would be prokaryotic. They would become prokaryotic, but they're right there at the edge of it, right? And red blood cells, they also they do not use for cell respiration. It's an anaerobic process. It's not aerobic. That's why they carry oxygen through it. They have to carry oxygen. Now, glycolysis. Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Okay? This is super important. Why? Why is glycolysis? I want to hear from one of you two, or those that are on here. I don't know how many people are on here. I apologize. I just saw 2 people comment. If you're not able to comment, please subscribe. It allows you to comment. It allows you to get and know when we are going live and a few other things. So please subscribe to the channel so you know when we're going live again, and you can actually chime in. So please, why is glycolysis so important? Why would you think it's important for us? Why would glycolysis be so important to understand? Because it's super important. Why would you think that? Please put that in the chat as we move forward. All right. So why would that be important? Glycolysis. Anyone? This thing has been acting up. I'm gonna have to get a new stand, because this stand has been. It might just be me kicking it, but it's been acting straight up. All right. So I'm gonna move to a different screen real quick. All right. So glycolysis, right? Let me do something real quick. There we go. I needed to do that. This up here. This. There we go. All right. So glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Nearly all living organisms can carry out glycolysis as a part of their metabolism. Okay. So why would glycolysis be so important for us to understand? This is super important. The process does not use oxygen. Now, this is what I want you to get. The process does not use oxygen and is therefore anaerobic. Processes that use oxygen are called aerobic. Glycolysis takes place in the cytoplasm of both prokaryotic and eukaryotic cells. All beings go through glycolysis, right? Now, both prokaryotic and eukaryotic cells. Glucose enters heterotrophic cells in two ways. Okay? Now, through secondary active transport, in which the transport takes place against the glucose concentration gradient, and through a group of integral proteins called GLUT proteins, also known as glucose transporter proteins. These transporters assist in the facilitated diffusion of glucose. I'm going to ask again, why is glycolysis so important? Why is it important for us to understand this? Glycolysis begins with the 6 carbon ring shaped structure of a single glucose molecule and ends with 2 molecules of 3 carbon sugar called pyruvate. Okay? So, very simple. This is how glucose starts off. Right? Now remember, everything has to be broken down to glucose, right? For the most part. So because of that, starch has can have up to 2 million strands of glucose. Right? So that's a lot. But everything has to be broken down to glucose. So it starts with this 6 ring structure, right? So what do we see here? Hydrogen all over the place, oxygen all over the place, right? And we see carbon all over the place. Hydrogen, oxygen, and carbon. This is what? What is glucose? Glucose is what for us? Glucose is what for us? I'm really waiting for an answer because I want somebody to answer that. Glucose is what for us? Can I answer? Please subscribe to the channel. Please put an answer in. What is glucose for us? Can you all hear me? I want to make sure y'all can hear me. Maybe you can't hear me. Or maybe you're that far behind me. I'm not sure. But glucose is what for us? I don't know if y'all can even hear me. I'm just keep going. I know it's recording, so it should be good. All right, so glucose is what for us? Glucose is food for us, right? So if glucose is food for us, right? Therefore, we need to understand this. So glucose then goes through glycolysis, which becomes 2 pyruvates. It's energy, sugar. Okay, great. So it's energy and sugar, right? It's food. This is what our food is. glucose, fructose. Those are our food. And what's going to end up at the end of glycolysis is 2 pyruvates. Y'all way behind me, I guess. Y'all must be way behind me. All right, so it's 2 pyruvates. That's the only thing I don't like about YouTube is that y'all end up so far. Sometimes y'all be so far off. I can't have a full conversation with you like I can with Zoom. Glycolysis consists of 10 steps divided into 2 distinct halves. Yeah, glycolysis consists of 10 steps divided into 2 distinct halves, right. The first half of the glycolysis is also known as the energy requiring steps, meaning this takes energy in order for it to take place. It takes energy in order for this to take place. So the first half of the glycolysis is also known as the energy requiring steps, okay? This pathway traps the glucose molecule in the cell and uses energy to modify it so that the 6-carbon molecule can be split evenly into the 3 carbon molecules. The second half of glycolysis, also known as energy releasing steps, extracts energy from the molecules and stores it in the form of ATP and NADH, the reduced form of NAD. Okay? So it extracts energy from the molecules and stores it in the form of ATP and NADH. Okay? Super important. So the first half of glycolysis, right, is here. 1, 2, 3, 4, 5. These are the first steps. So it goes from glucose to glucose 6 phosphate. It breaks it down. It takes ATP. It uses this really. This is an enzyme called hexokinase, right? Hexokinase is a enzyme. All right. When you see this A's on the end, that's an enzyme. Okay. So glucose, they're going to take ATP. It's going to take ATP and downgrade it kind of like to ADP. But it's going to take an ATP to do it. So you're going to lose an ATP at this first half. And then what's going to happen is going to create this glucose 6 phosphate. Okay? This is the phosphate right here. This is the glucose 6 chain right here. Okay? This is glucose total. This is stable glucose. This is a stable form. It's going to knock this into an unstable and add phosphate to it. Okay, to pull the energy so that it can like pull it. All right, then it's going to go and do the same. And then it's going to take this and it's going to turn this into fructose 6 phosphate. This is where fruit comes in. Really. Fruit will in the liver will be turned into glucose. So it'll come through this process. But 50% of fruit is not turned into glucose. It will come in through here. Right? Now, it's going to take another ATP, right, of energy, and it's going to create fructose 1 and 6 biphosphate. Okay? Glucose 1 biphosphate and 6 biphosphate. See them? Going to create that. Then what it's going to do is split them, right? It's going to take the fructose biphosphate aldose and it's going to split them in the dihydroxyacetone phosphate and glyceraldehyde. Glyceraldehyde 3 phosphate. Okay, glyceraldehyde 3 phosphate. It's going to split them here, just like this. You see the phosphates on each one. And then what's going to happen is it's going to move to the next phase. So let's read about these phases and totally understand them. Okay. Now, the first step in glycolysis is catalyzed hexokinase, an enzyme with broad specificity that catalyzes the phosphorylation of 6 carbon sugars. In other words, it's going to take these and split them. That's all it's saying. Really, just keep it simple. Hexokinase phosphorylates glucose using ATP as the source of the phosphate, producing glucose 6 phosphate, a more reactive, see that means unstable, more reactive form of glucose. This reaction prevents the phosphorylated glucose molecule from continuing to interact with the proteins. Okay? It stops it. And it can no longer leave the cell because the negatively charged phosphate will not allow it to cross the hydrophobic interior of the plasma membrane. And remember, when we talk about positive and negative, this is not good and bad. This is positive and negative in science. It's different. All right, it's charges, not morals. So this right here is where we're at right here. It creates this, what it's saying. Okay. Now it's going to go through another process after that. Okay, let's continue. And so it's saying that it won't allow it to leave the cell, right? Now, so it's in the cell because it needs to use it now. Step 2, in the second step of glycolysis, an isomerase, which is another enzyme, converts glucose 6-phosphate into one of its isomers, fructose 6-phosphate, okay? Which is what the body is going to use to create energy, fructose, right? An isomerase is an enzyme that catalyzes the conversion of a molecule into one of its isomers. This change from phosphoglucose to phosphofructose allows the eventual split of sugar into 3 carbon molecules. In other words, going from here to here is going to allow it to get closer to going to step 3, which is going to be fructose 1. The third step is the phosphorylation of fructose 6-phosphate catalyzed by the enzyme phosphofructokinase. A second ATP molecule donates a high energy phosphate to fructose 6-phosphate, producing fructose 1,6-biphosphate. In this pathway, phosphofructinase is a rate-limiting enzyme. It is active when the concentration of ADP is high, right? So if the ADP is high, that means that it's going to be active. Why? Because it needs to create ATP, right? It needs to create ATP, which is what? A triphosphate, 3 phosphates, not 1. Not 2, I mean. So it's going to be more energy, right? So if there is sufficient ATP in the system, when ADP is high, it is less active when it is active, right, because it wants to create ATP. It is less active when ADP levels are low and the concentration of ATP is high. Why? Because you have enough energy. So it can store the sugar. So ADP stays there because it's only a diphosphate acting as a resistor, not allowing as much through as the ATP would because you got enough energy. You got enough ATP. Thus, if there is sufficient ATP in the system, the pathway slows down. This is a type of end product inhibition since ATP is the end product of glucose catabolism. Okay? Right. So it works as a resistor if the ATP is high. If not, then you use ATP to create more ATP, right? But it's going to come out, it's going to double itself. And this is what we were talking about when we were talking about how cancer works. Because cancer, instead of moving into the metabolic state, into the mitochondria, where it goes through the Krebs cycle, it's going to continue trying to stay here, creating ADP. Even though the ATP could be sufficient, it's going to keep creating. Remember, I said it only creates 2 ATP? You remember that? It only creates 2 ATP over. That's cancer. Cancer stays in glycolysis. And remember, glycolysis is anaerobic. It doesn't move into the aerobic stage. It stays in the ADP. It stays, in the glycolysis stage, the anaerobic stage. where no spirit lives. It stays there. But remember, it can create it faster. Why? Because this process is only one stage process. It's actually two stages, but it's two stage versus going through an entire this and then a Krebs cycle to create energy. It keeps creating itself like this. Right? Thus, if there is sufficient ATP in the system, the pathway slows down. This is a type of end product inhibition, since ATP is the end product of glucose catabolism. Okay? Now, step 4, the newly added high energy phosphates further destabilize fructose 1 and 6. The fourth step in glycolysis employs an enzyme aldolase to cleave 1,6 biphosphate into 2,3 carbon isomers, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So let's look at that. So it's saying that right here, the fructose biphosphate aldose is going to break these up, bang, into this and into that. Got it? That's what it's going to do. All right, and I think there should be is that aldose or aldase? I think it's dase, but I'm not sure. I'll look at that again. They might have spelled that wrong on either side. But right here, it's splitting it into two, because it's going to be important that you understand these get split into two for the next stage of development. Okay? So we're here at step 4. It breaks it down. Yes, aldolase. That's what I thought. Okay, it's ACE. ASE. It's an enzyme, right? So now, the fifth, step 5. In the fifth step, an isomerase transforms the hydroxyacetone phosphate into its isomer, glyceraldehyde 3 phosphate. Thus, the pathway will continue with 2 molecules of a single isomer. At this point in the pathway, there is a net investment of energy from 2 ATP molecules in the breakdown of 1 glucose molecule. So in other words, it took an ATP here to break this down and an ATP here to break this down. But it did not take an ATP to break this down. Okay? But it did take 2 ATP to get there. All right? Now, I don't know if 50% of fruit comes through the glucose, but the other 50% comes through here. Now, I don't know if it takes an extra ATP for that to take place, because I have to look at the process to get from here to there. So it might be another ATP, but I'm not sure. But I know it skips this step for 50% of fruit. Right? So when you eat a piece of fruit, 50% goes into the liver, gets stored as glucose automatically. That's going to happen. This right here, and then when you release, but it's not being released. When it's released, then it's going to be used like this. But there's 50% of fruit that goes straight here to fructose, and then it's only using 1 ATP. So it might be taking up as much energy, and you might be getting more energy from it. But I'm not sure. I haven't found that yet. But I'll let you know. But that makes sense to me. But I have to look deeper into it to let you all know on that one. Okay? However, let's continue. Second half of glycolysis, energy releasing steps. So far, glycolysis has cost the cell 2 ATP molecules and produced 2 small 3 carbon sugar molecules, right? Which we saw here. These are the 2 small right here. This one, dihydroxyacetone phosphate and the glyceraldehyde 3 phosphate, right? Those 2. So now, both of these molecules will proceed through the second half of the pathway, and sufficient energy will be extracted to pay back the 2 ATP molecules, meaning it's going to get a bonus for spending the 2 ATPs, okay? So it'll be a payback the 2 ATP molecules used as the initial investments and produce a profit for the sale of 2 additional ATP molecules and 2 even higher energy NADH molecules. So it's going to produce 2 additional ATPs, then you're going to have 4 instead of 2, and then you're going to have NADH molecules. So here we go. So we got the glyceraldehyde 3 phosphate comes through here, right? And the dehydrogenase, right? It's going to come through here. This happens 2 times, not once, right? Because we got both of these. We got this one and we got this one. We got both of these. All right. So now what's going to happen is it's going to come through here, bang, and then it's going to go through twice. That's why you only have ADP twice here, but it's going to happen 2 times. It's going to produce NADH plus and NADHH plus H plus, right? That's hydrogen. That's the positive. Or I guess it's minus an electron. Or I guess it's an extra proton. I guess. I think that's what that means. I got to make sure. But NADH plus and NADHH plus. Plus phosphate, I guess that is. Yeah, that's plus phosphate. So, and then it's going to come down. It's going to be a 1,3-biphosphate glycerate, which looks like, again, fructose, like this was, right? Let's see. Where was that at? Here we go. See, look up fructose 1-biphosphate. This again is a biphosphate, 1 and 3 biphosphate right here. Okay. Then it's going to move through this process, ADP to ATP. So remember, when the ADP is high, then it's going to create an ATP, right? Then the 3-phosphate glycerate, right, is going to create this. Then a 2-phosphoglycerate is going to create that. Then you're going to lose a water molecule, right? And then phosphoenolipyruvate, right? PEP. This is a very important product right here. This product right here is extremely important, okay? Just for future reference, okay? And we see the phosphate here, phosphate, everything became phosphates from that stage, right? From this process. And then it's going to create, then pyruvate kinase, which is a enzyme, is going to produce pyruvate. So each one of these are a enzyme right here. So this right here, the glyceraldehyde 3-phosphate dehydrogenase, that's a enzyme, right? Splitting enzymes, I guess. These are splitting. So then we have the kinase enzyme here. We have a mutase enzyme here, an enolase enzyme here, and a kinase enzyme here. Okay, on the steps. Now, the 6th step in glycolysis oxidizes the sugar. Okay, it oxidizes it, extracting high energy electrons, NAD plus and NADH, which are picked up by the electron carrier, NADH plus, producing NADH. The sugar is then phosphorylated by the addition of a second phosphate group, producing 1 and 3 biphosphoglycerate. So no, that's not fructose. It's biphosphoglycerate itself. Note, the second phosphate group does not require another ATP molecule. Okay? So the second phosphate group does not require another ATP molecule. That's beautiful. Here again is a potential limiting factor for this pathway. The continuation of the reaction depends upon the availability of the oxidized form of the electron carrier, NAD plus. This is why this is so important. There used to be a supplement I used to deal with. I used to actually sell it, but it was a supplement that I used to take. I used to deal with this years ago, and it was based in NAD. NAD was the key, and they considered NAD at this time, and I think they still do. They considered this like the issue with aging. In other words, they had 90 and 100-year-old men and women running decathlons and marathons and wrestling and doing martial arts and everything based on their NAD levels. So NAD is something extremely important for our bodies, right? which now makes perfect sense. It makes perfect sense. The continuation of the reaction depends upon the availability of the oxidized form of the electron carrier NAD+. Thus, NADH must be continuously oxidized back into NAD+. In order to keep this step going. This is a key factor right here. This might be why we all lacking energy. I don't lack it no more, but why I did lack energy. Let me say it that way, because I got so much energy now. I don't even get tired. Thus, NADH must be continuously oxidized back into NAD plus in order to keep this step going. If NAD plus is not available, the second half of glycolysis slows down or stops. Now, mind you, this is before it even has to go through the entire Krebs cycle, which we're going to talk about tomorrow. If that stops, you have no energy. This is a key point right here. This is a high point. Back in NAD plus to keep this step going. If NAD plus is not available, the second half of glycolysis slows down or stops. If oxygen is available in the system, the NADH will be oxidized readily, though indirectly, and the high energy electrons from the hydrogen released in this process will be used to produce ATP. So the more spirit in the body, it just told you, the more NADH. That's why I keep telling y'all, wherever spirit is not available, disease is going to be there. I'm telling you, this is real. Like, if oxygen is available in the system, the NADH will be oxidized readily, though indirectly, and the high energy electrons from hydrogen released in this process will be used to produce ADP. In an environment without oxygen, an alternate pathway, fermentation, can provide oxidation of NADH to NADH, but that's backup. And we don't want to ever stay in a negative situation because it's going to cause what? Problems. Because fermentation is going to create alcohol. And you know what that does, right? Step 7. In the 7th step, catalyzed by phosphoglycerate kinase, an enzyme named for the reverse reaction, 1,3-biphosphate phosphoglycerate donates a high energy phosphate to ADP. Okay? So in the 7th step, let's get there. Let's go there. So we went to 6. This was 6, right? NADH plus connects to the phosphate. NADH plus connects to a hydrogen plus, right? Allowing for the connection to the extra electrons, right? And we from oxygen, right? However, if the oxygen is not available, it will use fermentation instead. However, that cannot be good. That's not going to be good. That's good, right? So we want to make sure that happens. Now, it says ADP to ATP. It's taking an ADP, we're in 7. So it's taking an ADP and converting it into ATP. And this happens twice. So we're going to end up with 4 ATP instead of just 2 ATP like it looks like on here. Okay? Now, in the 7th step, catalyzed by phosphoglycerate kinase, an enzyme named for the reverse reaction, 1,3-biphosphate glycerate donates a high energy phosphate to ADP, forming one molecule of ATP. This is an example of substrate level phosphorylation. A carbonyl group of the 1,3-biphosphate is oxidized to a carboxyl group, and 3-phosphoglycerate is formed, meaning this is an alcohol process, I believe, but I'm not sure. Step 9, enolase catalyzes the 9th step. Oh no, we skipped step 8, right? In step 8, the remaining phosphate group 3-phosphoglycerate moves from the 3rd carbon to the 2nd carbon, producing 2-phosphoglycerate. Let's look at that. Next, the remaining phosphate group moves. See it move. So it moves from here, from the 3 to the 2, right? See it? So this is group 1, group 2, group 3. It went from group 3 to group 2, right? And so that's where it's at right now. So it's going to be, that's why they name it that. So the remaining phosphate group 3 phosphoglycerate moves from their 3rd carbon to the 2nd carbon, producing 2 phosphoglycerate, an isomer of 3-phosphoglycerate. The enzyme catalyzing this step is a mutase, a type of isomerase. Okay. Step 9, enolase catalyzes the ninth step. This enzyme causes 2-phosphoglycerate to lose water from its structure. This is a dehydration reaction resulting in the formation of a double bond that increases the potential energy in the remaining phosphate bond and produces phosphoenolpyruvate, which is PEP. Okay, PEP. PEP is very important. That's something that's super important. You see it. I see it all over the place when dealing with health. Okay, so it does that, right? It loses its water. It dehydrates it to make it strong, more sugary, basically, for what? So it can now give the energy that it needs to produce this ATP, this second ATP. And remember, it goes through this process twice, so it's going to produce a third and a fourth ATP also. That's why this is broken down into 2, 1, 2, right? And it's going so they could drop down into here. Now just think. Do you know think of how much work? I just want you to think about how much work went into actually making this a reality. All we're doing is studying what was created. You understand what I'm saying? You see how great creation? It's just ridiculous. It's beautiful. Like, I mean, this is masterful. You understand what I'm saying? Step 9, enolase. I gotta say hallelujah, because this is masterful. Enolase catalyzes the ninth step. This enzyme causes 2-phosphoglycerate to lose water from its structure. This is a dehydration reaction resulting in the formation of a double bond that increases the potential energy in the remaining phosphate bond and produces phosphoenolpyruvate. Okay? Now, the last step in glycolysis is catalyzed by the enzyme pyruvate kinase. Okay? The enzyme in this case is named for the reverse reaction of pyruvate's conversion into PEP and results in the production of a second ATP molecule by substrate level phosphorylation and the compound pyruvic acid, or its salt form, pyruvate. Many enzymes and enzymatic pathways are named for the reverse reaction since the enzyme can catalyze both forward and reverse reactions. Right? So enzymes can go backwards and forwards is what it's saying. But anyway, it goes forwards here, I believe. And it's going to create ATP. And it's going to create this final form of sugar called pyruvate that is ready now to be moved to the mitochondria. All right? So it's ready to be moved to the mitochondria. So the outcomes of glycolysis, okay? Glycolysis starts with glucose and ends with 2 pyruvate molecules, a total of 4 ATP molecules and 2 molecules of NADH. 2 ATP molecules were used in the first half of the pathway to prepare the 6-carbon ring for cleavage. So the cell has a net gain of 2 ATP molecules and 2 NAD molecules for its use. So we gained 2 NADH and 2 ATPs. Okay? Because it gave us 4. We used 2. We created 4 and 2 NADH. Okay? So because of that, we end up with 2 extra NADH and 2 ATP. When the body is properly metabolizing sugar or glucose or fructose, right? When it's properly doing that, then it's creating this. When it's not properly doing it, we're not getting this. And this is where our problems are coming in. If the cell cannot catabolize the pyruvate molecules further, it will harvest only 2 ATP molecules from 1 molecule of glucose, which is what we were talking about with cancer, remember? Remember what we were talking about with cancer? And remember they were saying it was more efficient, but the only reason it was quicker for the cancer cells to use it, yes. However, I was saying it was less efficient because it did not produce the 4. Right? So because of that, that's where the problem came in. And then it doesn't even go through. It doesn't go through the Krebs cycle. So it needs to do that. Okay. So if a cell cannot metabolize, that's why I said earlier. It said what's the problems that are caused by the mitochondria acting up. It said cancer. You got it? So we gotta make sure that we're keeping our cellular respiration clean. That's why that balance oil is so important. I cannot stress it enough. It's that important. If the cell cannot catabolize the pyruvate molecules further, it will harvest only 2 ATP molecules from 1 molecule of glucose. Mature mammalian red blood cells are not capable of aerobic respiration. This is very important, okay? So, our blood cells, the mature ones, are not capable of aerobic respiration, the process in which organisms convert energy in the presence of oxygen, and glycolysis is their sole source of ATP. If glycolysis is interrupted, these cells lose their ability to maintain their sodium-potassium pumps, and eventually they die. Okay? The last step in glycolysis will not occur if pyruvate kinase, the enzyme that catalyzes the formation of pyruvate, is not available in sufficient quantities. In this situation, the entire glycolysis pathway will proceed, but only 2 ATP molecules will be made in the second half. Right? This is very important. The last step in glycolysis will not occur if pyruvate kinase, the enzyme that catalyzes the formation of pyruvate, is not available in sufficient quantities. So we need to find out what? Where do we get pyruvate kinase, don't we? We need to know that. Because if not, our body is not going to kick into the second half. We're going to have no energy, or very low energy. Right? That's no different than cancer. In this situation, the entire glycolysis pathway will proceed, but only 2 ATP molecules will be made in the second half. So your body's doing the same amount of work, but only getting half of the benefit. Thus, pyruvate kinase is a rate-limiting enzyme for glycolysis. We got to have that. Pyruvate kinase. Let's look that up. Shoot, I want to see. Let me see that. What foods do we get pyruvate kinase from? There isn't much information about what foods directly provide pyruvate kinase, but there's some related information about pyruvate kinase diet. Here we go. Dietary control. A high carbohydrate diet, insulin, and fructose can increase the activity of liver L-type pyruvate kinase. Right? So there you go. So fructose is going to be key. Why? Because fructose goes straight to the liver. Starvation. Starved rats have decreased liver L-type pyruvate kinase activity. Pyruvate kinase is an enzyme that converts phosphoenol pyruvate to pyruvate in the final step of glycolysis, right? So that's an alcohol process. A process that produces energy. That's fermentation, right? That produces energy. There are 4 isozymes of pyruvate kinase in vertebrates. L expressed in the liver, R expressed in the erythrocytes, M1 expressed in muscle and brain, and M2 expressed in early fetal tissue and most adult tissues. Pyruvate kinase deficiency is caused by mutations of the PKLR gene, which provides instructions for making the enzyme in the liver and red blood cells. I gotta look deeper into that. That's super important. Super important. In summary, glycolysis is the first pathway used in the breakdown of glucose to extract energy. It was probably one of the earliest metabolic pathways to evolve and is used by nearly all of the organisms on Earth. Glycolysis consists of two parts. The first part prepares the 6-carbon ring of glucose for cleavage into the 3-carbon sugars, right? Which we saw. In other words, it's preparing for step 10, right? It's preparing for step 10 pyruvate by creating these two sugars in step 5. So it's going to go from here to here for preparation, okay? Which takes energy, right? Then what does it say? Extract energy is the pathway used to break down glucose. It was probably one of the earliest metabolic pathways to evolve and used nearly on all organisms on Earth. Glycolysis consists of two parts. The first part prepares the 6-carbon ring of glucose for cleavage into the 3-carbon sugars, which we just talked about. ATP is invested in the process during this half of energize the separation. Right, we lose 2 ATPs to create the ADP that's necessary in order for us to move forward to get to the preparation by breaking down into the 3-carbon. Right. The second half of the glycolysis extracts ATP and high energy electrons from the hydrogen atoms and attaches them to ADT plus. 2 ATP molecules are invested in the first half, and 4 ATP molecules are formed by the substrate phosphorylation during the second half. This produces a net gain of 2 ATP and 2 NADH molecules for the cell. And this is a quick breakdown, right? It's harder to see it this way, but we see that it's going from glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1 to 6-BP. Then it's going to go down to the GADP, which I didn't see that, but DHAP, right? Going to break down here into those two. We're going to get the GAPDH here, right, at 6. Swinging around, this is where we're going to get the NADH as well as the the NAD plus and the NADHs, right? Then we're going to swing around here. It's going to go through its process here from the 1,3-PG, which is the phosphate glycinate, I think it is. Glycinate, yeah, I think that's what it is. And then it goes through the 3. Remember, the phosphate's on stage 3, and then it moves to the stage 2 phosphate, right? Where it moved, and it turns into the PEP, which is the pyruvate poly something. I can't remember how to pronounce it, but PEP, we know what that is. And then it becomes the pyruvate, which is necessary. And tomorrow we will kick into not tomorrow, because tomorrow we have the creation series. We'll be dealing with the formula for creation. This will be the step-by-step formula for creation we'll be going over tomorrow. So that will be either Sunday or Monday. We will have the pyruvate breakdown for you all. The mitochondria Krebs cycle breakdown, and how the mitochondria takes the pyruvate and creates the energy that we should be creating versus getting stuck here and not creating the energy that our body should be creating. Okay, so we need to understand this process because this is how we get energy. This is what keeps our body going. And remember, glucose is food for us, right? Fructose is food for us. All right. And when we understand this breakdown all the way from what I went through yesterday, hydrogen, how the earth and the world was created, all the way to this, now you can start to understand these breakdowns. So what do we have here? We got a bunch of hydrogen, oxygen, right? And phosphates, right? And if we look at it from here, we see the main piece, another main piece is carbon all through here. Carbon, 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 right? Carbon is holding it all together. So hydrogen is all out here, right? Carbon is holding it all together. Oxygen is key in the process. We see oxygen, carbon, and hydrogen. Very important. So what is hydrogen? Hydrogen is one. I call it the creation molecule or the creation atom because these are atoms. I call it the creation atom. Then we got carbon, right? Which is what? Carbon is 12. Carbon 12 is miracle because remember, until they could find beryllium 8, they had no idea how it was created, and beryllium 8 only lasts less than a second. And it must be able to fuse with helium in order to create carbon. So you have to have helium 4 plus beryllium 8 in order to create carbon 12. Right? Then that's carbon. That's a miracle that is even here. We need it. That's how important it is. And then oxygen is what? 16. Okay? So helium plus carbon equals oxygen. I believe it is. Yeah, it's helium plus carbon. Yeah. Right? So this is key. Okay? When they fuse, meaning that this is done. All of these products were made in the stars, minus helium. Even here, I mean, excuse me, minus hydrogen is also made in the stars now. So all of this was made by the sun, right? Like you don't. When they say you are nothing but stardust, you literally are. You literally are. All this is made in the stars. Isn't that amazing? You see how beautiful that is? It's beautiful, isn't it? It's perfection in the most beautiful way. That feels so good. So yeah, so thank you all for participating. Do we have any questions? Do we have any questions before we close out? Well, while we're waiting to see if there's any questions, I'm going to go ahead and play something real quick for you all. Welcome to my Patreon, where you might have come here from my YouTube or my Eventbrite where the videos expire within 30 days. Now you have access to my exclusive content forever once you join one of the tiers of my Patreon, okay? So we have the live stream health class, right, which you're going to get, but you're also going to get my on-demand health class. Remember, the health class is really 28 hours, but you in my live health class were able to receive 8 hours, 4 hours in session 1, 4 hours in the following session. You're also going to be able to get my ebook as well. So you can download my ebook, as well as those that saw me in person, you got a physical copy. You might have got it autographed, you might not. But however, you have a physical copy of my book as well. You got the live question and answers during my health class as well, and then the live chat during event. This whole health class portion is designed to give you the information necessary so that you can put the nutrients in your body that's going to allow your body to heal itself. Once you get your physical health in check, now it's time to move up a tier and get high with Daweed. Getting high with Daweed is basically a physical and mental, spiritual portion of the course. This is where you're going to receive my creation series, where you are able to create anything that you want in your environment, your space, or your reality whenever you want to. This is such a powerful course. I was actually watching it heal relationships, and I've seen people actually physically heal their bodies. This was before I was doing the physical health class. They were physically healing their bodies with their mind. And so it's an extremely powerful course. It's an extremely powerful portion and tier of the course as well. And you get everything in tier 1, which is the physical health class, plus you get the Getting High with Daweed creation series, okay, as well. You're also going to get my daily meditation videos here, which are very important to watch, right? You can actually physically heal yourself with just body meditation. Okay, so you're going to get my spiritual meditation, my physical meditation, and my mind meditation videos inside of this tier. Now, once you do that, or even before that, if you have a serious chronic problem, or you're not able to understand and be able to physically heal yourself just by watching my videos, then you can walk with Daweed. And walking with Daweed is very important. This is for the serious only. And I put together a tailored health system just for you. You have access to me where you can call me, you can email me, you can talk to me, and I will, or Zoom with me face to face, therefore that I will be able to help you and walk you through your physical ailment, mental ailment, or spiritual ailment, and walk you through that process. This is extremely powerful because it's not just about feeling better, it's about feeling normal again like you did when you were a child. No matter which tier you choose to start with, I'm looking forward to working with you. Peace. All right. So if you have any questions, there's no questions on the live. So if you have any questions later, please make sure that you enter them in the comments. I answer all questions that are placed in the comments. So feel free to leave them. I appreciate it. Thank you and have an outstanding day. See you at the top of your health journey. Peace. For the rest of this video and more videos like these, you can join us at patreon.com/health class. Again, that's patreon.com/health class. And join one of our paid tiers. Any of them will do. And you join one of those tiers, you can watch the rest of these videos, as well as all the other videos that I produced, including my former health classes, which is 4 to 8 hours of information that can help you on your health journey to the top. All right. So I look forward to sharing that adventure with you. I look forward to seeing you at the top of your health journey. Peace.